Today we're going to start talking about DNA testing, but first I want to remind you about some features about DNA in general and DNA polymerases. You've heard all this before, but a little gentle reminder probably wouldn't hurt before we jump in to PCR and other types of reactions. Okay, so DNA, I like to think about the different kinds of DNA in order to avoid confusion. The types of DNA include, of course, genomic DNA from an organism. Right? This is natural DNA that you would get when you acquired cells from a natural organism. It's huge, very large, complex, double-stranded, and even though the DNA gets broken into many pieces in the process of being isolated, still the pieces are many thousands of base pairs long. We've got 3 times 10 to the 9th base pairs in our haploid genome, so that's all going to be there when you make a DNA preparation from an organism. The opposite extreme is synthetic DNA oligonucleotides. These are made by machine, and they're made to order for whatever purpose the experimenter has in mind. So the sequence has been predetermined by whatever purpose they're being made for. And they're very short, only 20 or so nucleotides many times, up to 100 actually, really. And any sequence can be made. Importantly, they are single-stranded. You can make double strands out of them by making the complementary ones and mixing them together. But as they come off the machine, these are single-stranded molecules. And any sequence can be made. Okay, in addition, there are more synthetic types of DNA, or artificial, shall I say, because they're synthesized in vitro using oligonucleotide primers, for example. We saw cDNAs when we looked at our hemoglobin sequence, which is a copy of an mRNA sequence. So that cDNA, it does not exist in nature because the mRNA, of course, had its introns removed before becoming an mRNA. So the cDNA has essentially the exonic sequences and no introns. So it doesn't resemble its genomic DNA copy at all because it's missing all those important sequences. Okay, 2 strands of DNA, of course, are complementary and antiparallel. Okay, the complementary idea is very familiar. G pairs with C, A pairs with T. Okay, the polarity issue is a little bit more hard to understand, and people sometimes don't quite get it, so I'm going to remind you. Okay, so the 2 strands run in opposite directions. So polarity is not polarity in the chemical sense when we think about hydrophobic or something. It has to do with simply the direction of the molecule. Okay, and you can see that the 2 strands are running in opposite directions. Okay, the easiest way to think about this is to look at the sugar and orient to that. I like to think of the 5-carbon, which is sticking up from the sugar, as being the top or the beginning of the sugar, and the 2 to 3-carbon portion as the bottom. So then you can view the nucleotide as having direction from the 5' to the 3', top to bottom. Okay, so I'm drawing this picture. I've got a little image here of the 5' to 3' direction of the sugar, and then a single-stranded molecule in which those sugars are linked by, of course, the phosphates. So each sugar has a direction, so this molecule itself as a whole has a direction given by the direction of the individual sugar. So representing that here, I have 5', 3', 5', 3', etc. running down. So each one has a direction, so if we just link those together, we make a molecule which is overall running in the 5' to 3' direction relative to the sugars. Okay, so if you put those 2 together, you have to recognize that the 2 are antiparallel in that the sugar-phosphate backbone is running in the opposite direction on the opposite strand. The first strand on the left is running 5' to 3'. We can see the 5-carbon stuck at the top and running successively down the line till we have a free 3' hydroxyl on the bottom. Importantly, the other strand has the opposite polarity. It's literally standing on its head compared to its complement. So we have now the 5' carbon, the 5' end, at the bottom of the molecule and going successively up towards the top, running 5' to 3' in that direction. So the 2 halves of the molecule have opposite polarity, and that's something you have to keep in mind when you start looking at DNA sequences. Okay, so going on to hybridization, etc., the bonds holding the adjacent nucleotides together in a single strand are covalent, of course, the phosphodiester bonds, etc. These are not easily broken by mild conditions. However, the bonds holding the two strands together are non-covalent. They're hydrogen bonds, etc., and these are easily broken by conditions like high pH, high temperature, etc. So as a result, the two strands can be easily separated from each other or denatured under conditions that are not going to disturb the information content of either one of the strands. They remain intact. Okay, so separated strands, of course, can be reannealed to each other, or, and more importantly, for experimental reasons, they can be hybridized or annealed to complementary DNA molecules that we provide. So instead of going back with their own natural complements, we're going to use single-stranded DNA for that purpose. So the single-stranded oligonucleotides will sometimes serve as primers for DNA replication or probes to detect their complements or whatever. But the idea is, of course, we can provide a complement to any sequence that we want in the form of these small oligonucleotides for whatever the purpose may be. So here's an example. We have a small oligonucleotide which is labeled at its 5' end with a fluorescent compound, and it's trying to find its complement, which is the target sequence in the DNA. Now, this isn't the best representation because that target would actually normally be in a much larger molecule. This is just a representation of the actual hybridization. So in solution, the labeled oligo floats around, and when it encounters its complement, it will base pair in a reasonably stable way. And that will be unique to that exact sequence. So it can find its complement even in a very complex mixture of molecules. Okay, so note the little dot. That means we can see that later, right? Whether, like, for example, with the fish experiment we talked about a while back, the probes themselves were labeled, and that allowed us to see them in the microscope. So that very frequently you will have your molecules labeled in some way that you can figure out where they were or what they did after the fact. Okay, so now we need to talk a little bit about DNA polymerases, synthesis or replication. The word synthesis is usually used when you're discussing an in vitro situation like this, rather than replication. Okay, DNA polymerases, of course, copy a template DNA to produce a new strand, which is complementary to the template. DNA polymerases also require a primer that's already base paired to the template. Now, in cells, during normal in vivo replication, the primer is provided by primase as replication proceeds. In vitro, we're going to be using specific primers to target the synthesis to the region that we want to copy, and that's what our primer is going to do for us. Okay, the primer will bind, and the polymerase will then add on nucleotides to the 3' ends of the new molecules, moving in a 5' to 3' direction on the new molecule. Okay, I've diagrammed this. This is a little picture of a small piece of DNA which has been denatured, so the two strands are separated from each other. And importantly, what we've done here is we've bound a little oligonucleotide primer represented by the 5' to 3' with a little OH on it. It's base paired to a template. I'm not showing the base pairs here, but it is base paired in such a way that that 3' OH is available to serve as a beginning point for DNA polymerase to come along and copy that template. So the first nucleotide in the template is a C, so we're going to look for a DGTP for the polymerase to then add on to that 3' OH. Okay, now we've added on to our 3' OH, providing a new place for polymerase to add. The next nucleotide in the template is an A, so we're going to look for a DTTP to complement that, and then polymerase will they be able to use that DTTP as something that it can then add to the next nucleotide. So this happens in a sequential manner until you get to someplace else, and you're going to run your way all the way across this particular template, making a new strand. So our first application of this idea will be PCR, which is one form of in vitro DNA synthesis, and it has certain requirements and behaves according to all the rules we know about DNA polymerases. There needs to be a template, which we will provide. We need deoxyribonucleotide triphosphates to create material to create the new strand. We need a DNA polymerase. Now, the requirements here are kind of picky because the polymerase that's used has to be very heat stable, because the whole process is going to involve heating and cooling and heating and the enzyme has to be able to put up with very high temperature for this to be successful. And of course, you need your buffer and salts, whatever polymerase likes. Importantly, we need primers, 2 of them, 1 for each strand. Now these primers are going to define where the DNA synthesis will start. It's going to direct the polymerase to the place that we want to copy, not all those other places in the genome that we don't want to copy. So we're going to selectively synthesize the region that's defined by the binding of those particular primers to the strands. Okay, reminder, these are short, about 20 nucleotides, single-stranded oligo, DNA, not RNA, and they are specific to the region of interest. It will bind in such a way, we hope, that it's only going to bind one location in the template at a specific location. The other primer will bind the opposite end of the region we want to amplify, and they're going to be set up so that their 3' hydroxyls are pointing towards each other, so in the end we're going to synthesize the region between. Okay, PCR reactions, one example of in vitro DNA synthesis. So it's an in vitro synthesis reaction. It behaves according to the usual rules of DNA replication. Goes 5' to 3' in the new molecule, needs a template, primers, etc. It's different from others in that it makes an enormous amount of a particular DNA sequence by repeated rounds of DNA synthesis, not just one. And it's going to involve selective DNA synthesis in which the choice of our primers determines the sequence of the DNA to be copied. It's going to involve cycling among different temperatures to allow many, many rounds of reaction to occur. Okay, here's a diagram showing you the first cycle of a PCR reaction. It's a little deceptive because it doesn't show that there's lots and lots and lots of primers floating around. It looks like they're used up in the reaction, and they are used up, but there's always driven by molar excess, so don't worry about that part of it. Okay, so we're starting off with a double-stranded region of DNA that we want to amplify. The first step is a heating step, which separates the 2 strands, and that's usually done to 94 or so degrees to separate the strands. The reaction is then cooled down to allow the primers to actually anneal to the template, because they certainly would not do that at the high temperature, usually around 55 or so degrees. Then the temperature is changed to an intermediate temperature of 70 or so, which is a good temperature for these heat-stable polymerases to work well. And of course, once that polymerases have latched on to the primer, they can then extend the primers using the deoxyribonucleotide triphosphates that we have that are sitting there in the reaction mix. Okay, this is an editorial opinion. The slide is a little annoying because it looks like somebody's standing there adding reagents. In fact, all the ingredients in the reaction are there in the first place. The template, the NTPs, etc. are all sitting there, including nobody's standing there adding buffer or adding anything. And the cycling, of course, is done by the machine. So it's like a lot easier than it looks, because once you get your reaction set up and push the button, the 30 cycles of synthesis happen pretty much all on their own. So that's just the first cycle. That's not going to give us too much. It's going to give us twice as much as we had in the first place. We've got a double-stranded DNA, denature, primers will anneal, then you extend the primers. So in the next round, the next cycle of PCR, you repeat that whole process. You denature the strands by increasing the temperature, cooling it to allow the primers to anneal, and then increasing the temperature a bit to allow the polymerase to extend. Then you do the whole process again, allowing for denaturation, annealing, extension, until 30 cycles have gone by, and you have a really enormous amount of amplification of product. Okay, so this is a picture of what you have, what specific products you end up at the end of several cycles of PCR. And the idea is, of course, you amplify it by the fact that each product is actually going to then serve as a template for the next step or the next cycle in the reaction. Okay, so the 2 strands that you get are complementary to each other, and they're fully base paired, and the primers are actually part of the product at the 5' ends of each strand of the products, and they all have the same sequence. Thought experiment: how many new strands of DNA will be synthesized if we use one primer and perform the synthesis 30 times? 30, 60, about 1 billion. Okay, the answer is 30. How many new strands of DNA will be present if we use 2 primers, one at each end of the region of interest, and repeat the synthesis 30 times? About 1 billion. Okay, the reason, of course, if you only use 1 primer, you can make only 1 strand, and every time you make another round of synthesis, you're going to be making the same strand. Whereas if you have 2 primers, you're making both strands, and that gives you the fact that you have 2 primers. New template strands are created with each round of replication. So we're going to make new strands, new templates, and that's where the chain reaction part of this comes from. There's more than one way to do DNA testing depending on what you're looking for. We might be looking for specific mutations in somebody's DNA, looking for a genetic disease, or in the example we'll talk about today, we're going to do paternity testing. So what actually happens is going to be different according to what you're trying to do or what you're trying to see. The first step in any case is to get DNA sample from the people. And then what you're going to do is decide what exact part of the genome you're trying to look at, which is to say what parts of the genome we want to amplify. So you need to design primers to amplify those, one at each end of the region of interest, whatever it is you're trying to see. So the DNA that you get from your PCR amplification will give you enough to look at by some technique or another. So the PCR in itself mostly isn't going to help you until you actually analyze the product in some way or another. You might just be looking for a single nucleotide change by sequencing, or you might be looking for the size change in the PCR product or whatever. But the first step is very often the PCR, and then apply some analytical technique to the product depending on what you're trying to see. Okay, so for paternity, the example is going to be DNA testing using STR loci, which is short tandem repeats, also called microsatellites. Okay, these loci are very strange, locus referring to location in the genome, and everybody has 2 alleles of each, one from mom and one from dad. So what makes them useful is they're very variable, and most people are heterozygous for many of these loci. And this will become clear in a sec when you see the pictures. The FBI long ago chose a group of such genomic regions for DNA testing purposes, and there's a huge amount of information about those particular regions and alleles and sequences. So the statistics are very well supported. So here's the original 13 CODIS core STR loci, which are used by the FBI. And they're shown represented on what chromosomes they come from or where they are. And they're given sort of odd-sounding names, like the D3S1358. Don't be intimidated by the name. It's just a way to talk about that particular location on that particular chromosome and the particular sequence that we're going to be trying to amplify. But the important point is they're spread all the way around the genome, and they're very well characterized. This is a newer version of the same thing. Typically 20 or so are used now, but there's, again, STR, the loci contain short tandem repeat sequences, and they're everywhere in the genome. Okay, so this refers to how they're named. 3S refers to the short arm of chromosome 3. Most of these are in non-transcribed region of the genome, but some are in introns. Importantly, all contain some kind of short repeat. I'll use GATA in the example shown. So we can define an allele by the number of GATAs on that particular allele of that particular locus. Okay, so here's an example of D3S1358. On the bottom, we have the sequences of the primers that are used, and they are italicized in the text diagram to show you where the synthesis reaction actually starts in a PCR. And you can see that they are on either side of this underlined region. The underlined region is the GATA repeats. This particular example of this particular locus has 18 repeats, which is to say it has 18 GATAs. But that number varies from one allele to another. This particular one has 18, but there's a lot of variation in the population as to how many actual ones they have. Right, so you might have there's going to be a range, in this case from 12 to 22, but we will describe the genotype of a person for this locus by whatever the number of repeats is. So for example, you might be 15, 17, meaning that one of your chromosomes has, chromosome 3 has 15 GATA repeats, and the other has 17. One is from mom and one is from dad. This is shown here maybe a little more clearly in that the allele on the top has 8 copies of this particular repeat. It's AATG is a repeat, and the primers are bound on both sides. So we're going to get a PCR product from that, which is going to go from the end of one primer to the end of the other primer. The allele underneath has only 7 copies of the repeats. Use the same primers, which bind in the same region outside the repeat region. So that PCR product is going to be 4 nucleotides shorter than the first one because it has only 7 AATG repeats versus 8. So that's how the alleles are designated, and that's how we will be able to identify them when we analyze these on our electrophoretic situation. Okay, this is from your Cell and Molecular book, and it illustrates the same point. We have a person who has, of course, 2 chromosomes, one from mom, 2 chromosomes, whatever it is, one from mom and one from dad. And that chromosome contains one of the STR loci that we use for genotyping. And we can see that the allele from dad has more copies of the green thing, which is the repeat region, than mom's does. So when we amplify that DNA with those primers, we're going to get a product, and we're going to get a product from mom's. But those 2 products are going to differ in size because mom's allele has fewer repeats than does dad's. So once we've done that, we have to look and see what we've got, and we do it by electrophoresis in this case, where we put the products, well, it's really in a capillary, not in a gel, but you get the idea. And we're going to run them, and the maternal one, of course, is a smaller product, so it's going to run faster than the paternal one. So those 2 allele products can be separated and then visualized in some way. Okay, so we need actually a very sensitive way to detect this because we're only looking for 4 nucleotides difference potentially between the products. So the regular gel, agarose gel electrophoresis won't work for us, but there are systems that really will that can basically resolve single nucleotide differences pretty easily. So our genotype can be determined by running it on that kind of a gel system or capillary system that's able to separate molecules with such an amazing degree of sensitivity. Okay, here's a picture. We don't just run the gel and stain it. The gel is actually run in such a way that the bands actually move away or move off or out of the capillary. And they are actually identified and seen by the fact that they're fluorescent. We've labeled the primers in the first place. And as they migrate past this laser, the fluorescence is activated, and that fluorescence is then detected and reported to the computer system as they go by. So it's not a static situation. The molecules are migrating, and they're being observed as they pass this detector system, and then reported to the software. Okay, so to make this work, you have to know the sizes that you're looking for, and allelic ladders are typically used for this purpose. So what the allelic ladder is basically, in this instance, the population variation goes from 5 repeats on the small side all the way up to 11. So anybody's going to have 2 of those bands at most, but the population, of course, has more. So when we genotype the person, we've run their PCR products through the gel, we'll be able to identify which allele it is by comparing it to the allelic ladder. The ladder goes for 5, 6, 7, 8, 9, 10, 11. So you look at which bands match the allelic ladder, and that tells you how many repeats on that particular individual has. So the person on the left has genotype 7 and 9 because their band runs with the 7 and with the 9. The person on the right has genotype 6, 8 because their bands run with the 6 on the ladder and the 8 on the ladder. So they can be genotyped by comparing to these ladders. Okay, in reality, you usually do several reactions at once, sometimes a really large number at once. That's called multiplexing. One way to do this is to have different colors of fluorescence so you can figure out which products came from which amplicon, etc. And the machine is able to call the allele according to the repeats that are present. All right, so here's one picture of a capillary situation where the products of 3 different loci are actually run together, and the peaks represent which actual allele is being seen by the instrument. So this particular person has alleles 10 and 11 for this locus, the D13S317 locus. This person, same person, has 8 and 10 from the D7S820 locus, etc. So these are all run together for efficiency, and the machine is able to detect which allele is present in the sample as compared to the allelic ladder. Okay, so for paternity testing, we have the fact that everybody has 2 alleles of each, one from mom and one from dad. Now, the 2 alleles may have the same size, but there's a lot of heterozygosity also. So we look at mom's alleles and decide which of the child's bands came from her, and the other one that did not come from mom must come from dad. So that's the obligatory paternal band. Sometimes it's not possible to tell, and that's why many loci are used simultaneously in order to be able to determine this. So for example, here's 2 examples, one where the man is excluded from being the father, and the other where the man is not excluded from being the father. They color-coded the paternal alleles for us on this one. Okay, so go by line, locus by locus. In each case, identify the obligatory paternal band, if possible. So what you do is you look at the child and decide which band must have come from the father because it's not present in the mother. So you can identify which came from the mom, and what the other one that's left over must be the one that came from the dad. Okay, so for example, the first line, CSF1PO, we're not going to learn too much from this locus because the child has alleles 12 and 13, mom has alleles 12 and 13, and dad has alleles 10 and 13. So the child could have gotten either 12 or 13 from the mom, we can't tell which, in which case, if he did get the 12 from the mom, then he could have gotten the 13 from the dad. So that's consistent with paternity. You can't exclude him on that basis. The next one, however, is different. We have the child has an 8 and a 14. Mom has an 8 and 11. So we know the child got one of his alleles from his mom, and therefore it must be the 8 because that's the one that's common to him and his mom. So that means that child's 8 came from his mom. The 14 must have come from the dad. So that's the obligatory paternal band. So we need to look at the possible dad and see if dad has that band. If he has that band, he's not excluded. But if he doesn't have the band, he is excluded because the child got it from his father. If that's his father, the father should have that band. So we look for the father. He has alleles 12 and 13, but no 14. So that's an exclusion. The child got his 14 from his dad, so this dad must not be dad because he doesn't have a 14 to give to his son. Now, if you go through the chart, there's many other examples of exclusions. I'll go to D18S51, for example. The child has 14 and 16. Mom has 16, so she's homozygous. She's got 2 16s. So we know that the child got the 16 from his mom. The 14, therefore, must have come from the dad. But this dad doesn't have a 14. He's homozygous for the 13. So he's excluded again on that basis. So if you go through the other alleles on the chart, you find several other exclusions from which we can conclude that this man is not the father of this child. Okay, in principle, it only takes one exclusion to eliminate a man as a father, but in practice, it's considered much better to get more than one exclusion. And with this many loci, that's a very likely situation to occur if he is not the father, in fact. Here's a case where there is no exclusion, and they've nicely color-coded this for us in identifying which alleles came from mom and which came from dad. So we go through all of these, and there's no exclusion for any of the loci. So the man is not excluded as a father. Now, you can't prove that he is the father. You just can't prove that he's not the father. But with population level statistics being as they are, the chances are, with that degree of match to this many loci, that in fact he is the father of the child. But it can't be strictly proven by this. Okay, so you should be able to go through each of these and see that what you see on the chart is completely consistent with this man being the father of the child. You can't do this unambiguously for all loci, but you can do it for many of them. So we will not go on to our next topic, which is allele-specific oligonucleotides today. We're going to save that for after Labor Day. Have a good weekend, everybody.